Study of cell or cell biology began with the discovery of cell by Robert Hooke in 1665. He prepared and studied thin sections of cork of dead plant material under his self-made compound microscope. He observed that cork is composed of minute honeycomb-like compartments, which he termed as cells. According to Hooke, cell is empty space bound by thick walls. Very little information was added to this idea in the following century. The work again started in the beginning of 19th century. Lawrence Oken, a German scientist in 1805, believed that all living beings originate from or consist of vesicles or cells. Jean-Baptiste de Lamarck in 1805 expressed similar idea and said nobody can have life of its constituent parts are not cellular tissues are not formed by cellular tissues. In 1831, Robert Brown reported the presence of nucleus in the cell. Due to this discovery, Hooke's idea about the cell as an empty space was changed. It was later established that cell is not an empty space. The German zoologist Theodor Schwann in 1839 and a German botanist Schleiden in 1838, working independently, came out with a theory called cell theory. They found that cell consists of three basic parts: nucleus, the fluid called cytoplasm that surrounds the nucleus, and an outer thin covering of membrane called plasma membrane. The cell wall, they said, was an additional structure present only in the plant cell. Keeping in view this definition of cell, the cells could be observed in plants as well as in animals. According to the cell theory, all living organisms are composed of cells or cell products. The cell theory is one of the most fundamental generalizations in biology. It has wide-ranging effects in all fields of biological sciences. Many details of the cells were studied, as a result of which the cell theory was extended. Rudolf Virchow, a German physician, in 1855 hypothesized that new cells were formed only by division of previously existing living cells. It was contrary to the idea of abiogenesis. Living things arise spontaneously from non-living things. One of the prevailing but controversial ideas about the origin of life at that time. Louis Pasteur in 1862 supplied experimental proof of Virchow's hypothesis by demonstrating that microorganisms, means bacteria, could be formed only by existing bacteria. Original cell theory and Virchow's hypothesis gave us the basis of working definition of living things. Living things are chemical organizations composed of cells and capable of reproducing themselves. August Weismann in 1880 said, all presently living cells have a common origin. The salient features of cell theory in its present form are: all organisms are composed of one or more cells. Number 2, all cells arise from pre-existing cells. Number 3, cell is a basic structural as well as functional unit of all organisms. In multicellular organisms, there is a division of labor among cells. Different cells are specialized for different functions. The function of organism as a whole is a result of sum of activities and interactions of different cells and different components of the cell. In animals, for example, muscle cells contract and relax, Nerve cells transmit impulses. Gland cells secrete. Red blood cells carry oxygen. And some stomach cells secrete gastric juice. Similarly, in plants, xylem cells conduct water and mineral salts from soil to the aerial parts of the plant. Phloem cells translocate food. Sclerenchymatous cells support to the plant. Chlorenchymatous cells carry out photosynthesis. Parenchymatous cells store surplus food. Meristematic cells produce new cells for growth and development of plant. As they perform different functions, they show great variations in shape and size. Despite the structural and functional diversity, plant cells as well as animal cells have common plan of organization. The human naked eye can differentiate between 2 points, which are at least 1 millimeter apart. This is known as resolution of eye. The resolution can increase with the aid of lenses. In a typical compound microscope, the resolution is 2 micrometer, which is about 500x that of the naked eye. A compound microscope is a typical laboratory microscope with at least different magnification powers. The typical ocular lenses could be 5x and 10x, but others also exist. Likewise, different types of objective lenses like 20x, 40x, 100x, etc. exist. The magnification power of microscope is determined by multiplying x value of ocular lens and x value of objective lens. Therefore, a microscope with 10x ocular lens and 40x objective lens will have 10 into 40, 400x magnifying power. The resolution will, however, remain the same, which is 500x that of the naked eye. The source of illumination in such microscope is visible light. In electron microscope, the source of illumination is a beam of electrons, and resolution of microscope ranges between 2 to 4 angstroms, which make it 500x greater than that of a compound microscope.